हमारा सेकंड यूनिट है स्ट्रक्चरल ऑर्गेनाइजेशन इन प्लांट्स एंड एनिमल्स. The description of the diverse forms of life on Earth was made only by observation through naked eyes or later through magnifying lenses and microscopes. This description is mainly of gross structural features, both external and internal. In addition, observable and perceivable living phenomena were also recorded as part of this description. Before experimental biology, or more specifically physiology, was established as a part of biology, naturalists described only biology. Hence, biology was amazing in terms of detail, while in initial reaction of a student could be boredom. One should keep in mind that the detailed description was utilized in the latter day reductionist biology, where living possesses drew more attention from scientists than the description of life forms, helpful in framing research questions in physiology or evolutionary biology. In the following chapters of this unit, the structural organization of plants and animals, including the structural basis of physiological or behavioral phenomena, is described. For convenience, this description of morphological and anatomical features is presented separately for plants and animals. Nathalie was born in Ukraine in 1898. They studied agriculture in Russia and Germany and received her doctorate in 1931 in the United States. She reported in her early publications that the curly top virus spreads through the plant via the food conducting or phloem tissue. Dr. Isau Plant Anatomy is published in 1954. Took a dynamic developmental approach designed to enhance one's understanding of plant structure and and of and an enormous impact worldwide, literally bringing about a revival was published in 1960. It was referred to as Webster's of Plant National Academy of Sciences, becoming the sixth woman to receive that honor. In addition to this prestigious award, she received the National Medal of Science from President George Bush in 1989. When Catherine Isau died in the year 1997, Peter Levin, Director of Anatomy and Morphology, Missouri Botanical Garden, remembered that she absolutely dominated the field of plant biology even at the age of 99. Morphology of flowering plant. The wide range of the structure of higher plants will never fall to fascinate us. Even through the angiosperms show such a large diversity of in external structure of morphology. They are also all characterized by presence of roots, stems, and leaves, flowers, and fruits. In chapter 2 and 3, we talked about classification of plants based on morphological and other characteristics. For any successful attempt at classification, and that understanding any higher plants for that matter any living organism. We need to know standards, technical terms, and standard definitions. We also need to know about the possible variations in different parts uh, and as adaptations of the plants to their environment. For example, adaptations to various habitats for protection, climbing, storage, etc. If you pull out any windweed, you will see that all of them have roots, stems, and leaves. They may be bearing flowers and fruits. Understanding part of flowering plant is the root system, while the portion above the ground forms the shoot system. Figure 5.1. The root. The majority of the dicotyledonous plants, the direct elongation of the radicle leads to the formation of primary root, which grows inside the soil. It bears lateral roots of several orders that are referred to as secondary, tertiary, etc. roots. The primary roots and its branches constitute the taproot system as seen in the mustard plant, which is located in figure 5.2a. In monocotyledonous plants, the primary root is short-lived and is placed by a large number of roots. These roots originate from the base of the stem and constitute the fibrous root system, as seen in the wheat plant figure 5.2b. In some plants like grass, monstera, and banyan tree, roots arise from parts of the plant other than the radicle are called adventitious roots. This is located in 5.2c. The main functions of the root system are absorption of water and minerals from the soil, providing a proper anchorage to the plant parts, storing reserve food material, and synthesis of plant growth regulators. Adventitious roots. Regions of the roots. The root is covered of the apex by a thimble-like structure called the root cap, which is located in figure 5.3. The region of root tip. It protects the tender apex of the root as it makes its way through the soil a few millimeters above. The root cap is the region of meristematic activity. The cells of this region are very small, thin-walled, and with dense protoplasm. They divide repeatedly. The cells proximal to this region undergo rapid elongation and enlargement and are responsible for the growth of the root in length. This region is called the region of elongation. The cells of the elongation zone gradually differentiate and mature. Hence, the zone proximal to region of elongation. This is called the region of maturation. From this region, some of the epidermal cells form very fine and delicate thread-like structures called root hairs. These root hairs absorb water and minerals from the soil. The stem. What are the features that distinguish the stem from a root? The stem is the ascending part of the axis, bearing branches, leaves, flowers, and fruits. It develops from the plumule of the embryo of a germinating seed. The stem bears nodes and internodes. The region of the stem where leaves are born are called nodes, while internodes are the portions between two nodes. The stem bears buds, which may be terminal or axillary. Stem is generally green when young and later often become woody and dark brown. The main function of the stem is spreading out branches bearing leaves, flowers, and fruits. It conducts water, minerals, and photosynthesis. Some stems perform the function of storage of food, support protection, and vegetative propagation. The leaf is a lateral, generally flattened structure borne on the stem. It develops at the node and bears buds in its axis. The axillary bud later develops into the branch. Leaves originate from shoot apical meristems and are arranged in acropetal order. They are the most important vegetative organs for photosynthesis. The typical leaf consists of three main parts: leaf base, petiole, and lamina, which is located in figure 5.4a. Stipule, petiole, leaf base, axillary bud, lamina. The leaf is attached to the stem by the leaf base and may bear two lateral small leaf-like structures called stipules. In monocotyledons, the leaf base expands into a sheath covering the stem partially or wholly. In some leguminous plants, the leaf base may become swollen, which is called the pulvinus. The petiole, petiole allow leaf blades to flutter in wind, thereby cooling the leaf and bringing fresh air to leaf surface. Part of the leaf with veins and veinlets. There is usually the middle prominent vein, which is known as the midrib. Veins provide rigidity to the leaf blade and act as channels and transport for water, minerals, and food materials. The shape, margin, apex, surface, and extent of incision of lamina varies in different leaves. Venation. The arrangement of veins and veinlets in the lamina of leaf is termed as venation. When the veinlets form a network, the venation is termed as reticulate, which is located in figure 5.4b. When the veins run parallel to each other within the lamina, the venation is termed as parallel, which is located in figure 5.4c. Leaves of dicotyledonous plants generally possess reticulate venation, while parallel venation is the characteristics of most monocotyledons. Types of leaves. A leaf is said to be simple when its lamina is entire or when incised. The incisions do not touch the midrib. When the incisions of the lamina reach up to the midrib, breaking it into a number of leaflets, the leaf is called a compound. The bud is present. A leaf is and the bud is present in the axilla of petiole in both simple and compound leaves, but not in the axil of leaflets of the compound leaf. The compound leaves may be of two types. In pinnately compound leaf, a number of leaflets are present on a common axis. The rachis, which represents the midrib of the leaf as in neem. The palmately compound leaves, the leaflets are attached at the common point, that is at the tip of petiole, as in silk cotton, China rose, guava, different types of phyllotaxy, alternate, opposite, word. Phyllotaxy. Phyllotaxy is the pattern of arrangement of leaves on the stem of branch. This is usually of three types: alternate, oppose, and fold, which is located in figure 5.6. In alternate type of phyllotaxy, a single leaf arises at each node in alternate manner, as in China rose. Alternate phyllotaxy, which is located in China rose. B. Opposite phyllotaxy, which is located in guava, and world phyllotaxy, which is located in Estonia. A pair of leaves arise at each node and lie opposite to each other, as in Calliostropis and guava plants. If more than two leaves arise at a node and form a whorl, it is called a whorl, as in Australia. The inflorescence of flower is a modified shoot wherein the shoot apical meristem changes into floral meristem condensed. The apex produces different successive nodes. Instead of leaves, when a shoot tip transforms into a flower, it is always solitary. The arrangement of flowers on the floral axis is termed as inflorescence, depending on whether the apex gets developed into a flower and continues to grow. Two major types of inflorescence are defined: racemose and cymose. In this racemose type of inflorescence, the main axis continues to grow. The flowers are borne laterally in an acropetal succession, which is located in figure 5.7. In cymose type of inflorescence, the main axis terminates in a flower, hence is limited in growth. The flowers are borne in a basipetal order. 5.7. The flower is the conducting. The flower is the reproductive unit in the angiosperms. It is meant for sexual reproduction. The typical flower has four different kinds of whorls arranged successively on a swollen end of the arranged successively on the swollen end of the stalk and pedicel, called thalamus and receptacles. These are calyx, corolla, androecium, and gynoecium. Calyx and corolla are accessory organs, while androecium and gynoecium are reproductive organs in some flowers like lily. The calyx and corolla are not distinct and are termed as perianth. When a flower has both androecium and gynoecium, it is bisexual. The flower having either only stamens or only carpels is unisexual. In symmetry, the flower may be actinomorphic, radial symmetry, or zygomorphic, bilateral symmetry, when a flower can be divided into two equal radial halves in any radial plane passing through the center. It is said to be actinomorphic. For example, mustard, datura, chili. When it can be divided into two similar halves only in one particular vertical plane, it is irregular. It is if it cannot be divided into two similar halves by any vertical plane passing through the center, as in kana. The flower may be trimerous, tetramerous, or pentamerous. When in floral appendages are in multiple of 3, 4, and 5 respectively. Flowers of with bracts, reduced leaf found as the base of the pedicel are called bracteate, and those without bract, ebracteate. Position of floral parts of thalamus, hypogynous, B and C perigynous, and D epigynous. Hypogynous, perigynous, and epigynous. Based on the position of calyx, corolla, and androecium in respect of the ovary of thalamus, the flowers are described as hypogynous, perigynous, and epigynous. The flowers are described as hypogynous, perigynous, and epigynous, which is located in figure 5.9. In the hypogynous flower is the gynoecium flower. The gynoecium occupies the highest position, while the other parts are situated below it. The ovary in such flowers is said to be superior. For example, mustard, China rose, and brinjal. If gynoecium is studied, situated in the center, and other parts of the flower are located on the rim of the thalamus, almost are the same level. It is called the perigynous. The ovary here is said to be half inferior. For example, plum, rose, peach. In epigynous flowers, the margin of thalamus grows upward, enclosing the ovary completely and getting fused with it. The other parts of flower are rise above the ovary, hence the ovary is said to be inferior, as in flowers of guava and cucumber, and the ray florets of sunflower. Parts of a flower. Each flower normally has 4 floral whorls for vise, calyx, corolla, androecium, and gynoecium. Calyx. The calyx is the outermost whorl of the flower, and the members of called sepals. Generally, sepals are green, leaf-like, and protect the flower in the bud stage. The calyx may be gamosepalous, sepals united, and polysepalous, sepals free. Corolla. Corolla is composed of petals. Petals are usually brightly colored to brightly colored to brightly colored to attract insects for pollination, like calyx. Corolla may also be gamocephalous, petals united, or polycephalous, polypetalous, gamopetalous, and polypetalous, petals free. The shape and color of corolla vary greatly in plants. Corolla may be tubular, bell-shaped, funnel-shaped, and wheel-shaped. Aestivation. The mode of arrangement of sepals or petals in floral bud. Floral bud with respect to the other members of the same whorl is known as aestivation. The main types of aestivation are valvate, twisted, imbricate, vexillary, which is located in figure 5.11. Aestivation. When sepals or petals in the whorl just touch and one another at the margin. Without overlapping, as in calotropis. It is said to be velvet if one margin of the appendage overlaps that of the next one, and so on. It is ladyfinger and cotton. It is twisted. If the margins of sepals and petals overlap on one another, but not in particular direction as in cassia and gulmohar, the activation is called imbricate. In pea and bean flowers, there are five petals. The largest standard overlaps the two lateral petals wings, which in turn overlap the two smallest anterior petals. Keel. This type of aestivation is known as maxillary or papilionaceous. Androecium. Androecium is composed of stamens, each stamen which represents the male reproductive organ, consists of a stalk or a filament and an anther. Each anther is usually bilobed, and each lobe has two chambers. The pollen sacs. The pollen grains are produced in pollen sacs. The sterile stem is called a seminar. Stamens of flower may be united with other members such as petals or among themselves. When stamens are attached to the petals, they are epipetalous, as in brinjal, or epiphyllous when attached to the perianth, as in the flowers of lily. The stamens in the flower may either remain free, polyandrous, or may be united in varying degrees. Then stamens may be united into one branch or one bundle, monoadelphous, as in China rose, and two bundles, diadelphous, as in pea, or into more than two bundles, polyadelphous, as in citrus. There may be of variation in the length of filament within a flower, as in salvia and mustard. Gynoecium. Gynoecium is the female reproductive part of flower, as in made and is made up of one or more carpels. The carpel consists of three parts, namely stigma, style, and ovary. Ovary is the enlarged basal part on which lies the elongated tube. The style. The style connects the ovary to the stigma. The stigma is usually at the tip of the style and is the receptive surface of pollen grains. Each ovary bears one or more ovules attached to the flattened cushion-like placenta. When more than one carpel is present, they may be free, as in lotus and rose, and are called apocarpus. They are termed syncarpus when carpels are fused, as in mustard and tomato. After fertilization, the ovules develop into seeds and the ovary matures into a fruit. Placentation. The arrangement of ovules within the ovary is known as placentation. The placentation are of different types of, namely marginal, axial, parietal, basal, central, and free central. Free central, which is located in figure 5.12. The marginal placentation, the placenta forms a bridge along a ventral structure of the ovary, and the ovules are born on this ridge. On this ridge, ovules are attached to it in multilocular ovary, and the placentation is said to be exile as in China rose, tomato, and lemon. In parietal placentation, the ovules develop on the inner wall of the ovary or chambered due to the formation of false septum, for example, mustard and argemone. When the ovules are born on central axis and septum are absent, as in dianthus and primrose, the placentation is called free central. The basal placentation, the placenta develops at the base of ovary and single ovule is attached to it as in sunflower marigold. The fruit is a characteristic feature of flowering plants. It is of mature and ripened ovary developed after fertilization. If the fruit is formed without fertilization of the ovary, it is called a parthenocarpic fruit. Usually, the fruit consists of whole and pericarp. The seeds and the pericarp may be dry or fleshy. When pericarp is thick and fleshy, it is differentiated into the outer epicarp, the middle mesocarp, and the inner endocarp. A mango In mango and coconut, the fruit is known as the drupe, which is located in figure 5.13. May develop They develop from monocarpellary superior ovaries and are one-seeded. In mango, the pericarp is well differentiated into an outer thin epicarp, the middle fleshy edible mesocarp, and the inner stony hard endocarp. In coconut, which is also a drupe, the mesocarp fibrous, which is located in figure 5.12. Types of placentation: marginal, axial, parietal, free central, and basal. Marginal, axial, parietal, free central, and basal. So यहाँ पर हम 5.13 फिगर for parts of fruits, the mango है, coconut है, epicarp, mesocarp, seed, endocarp. The seed. The ovules after fertilization develop into seeds. A seed is made up of a seed coat and an embryo. The embryo is made up of a radicle and an embryonal axis and one as in wheat, maize, and two cotyledons as in gram and pea. Structure of dicotyledonous seeds. The outermost covering of the seed is the seed coat. The seed coat has two layers: the outer testa and the inner tegmen. The hilum is a scar on the seed coat through which the developing seeds were attached to the fruit. Above the hilum is a small pore called the micropyle. Within the seed coat is the embryo, consisting of an embryonal axis and two cotyledons. The cotyledons are often fleshy and full of reserve food materials. At the two ends of the embryonal axis are present the radicle and the plumule, which is located in figure 5.14. Structure of dicotyledonous seeds. In some seeds, such as castor and endosperm formed as the result of called endospermic seeds and plants such as bean, gram, and pea. The endosperm is not present in mature seeds, and such seeds are called non-endospermous. Structure of monocotyledonous seeds. Generally, monocotyledonous seeds are endospermic, but sesames, as in orchids, are not non-endospermic. Some in the seeds of cereals such as maize, the seed coat is membranous and generally fused with the fruit wall. The endosperm is bulky and stores food. The outer covering of endosperm separates the embryo by a proteinaceous layer called the scleron layer. The embryo is small and situated as in groove at one end of the endosperm. It consists of one large and shield-shaped cotyledon known as scutellum and a short axis with the plumule and a radicle. The plumule and radicle are enclosed in sheaths which are called coleoptile and coleorhiza respectively, which is located in figure 5.15 structure of monocotyledonous seed. First primary endosperm embryo, then a second seed coat, fruit wall, aileron layer, endosperm, scutellum, coleoptile, plumule, radicle, coleorhiza. Semi-technical description of a typical flowering plant. Various morphological features are used to describe a flowering plant. The description has to be brief in a simple and scientific language are presented in a proper sequence. The plant is described beginning with its habit, vegetative characters, roots, stems, and leaves, and then floral characters, inflorescence, and flower parts. After describing various parts of plant, the floral diagram and floral formula are presented. The floral formula is represented by some symbols in the floral formula. BR stands for bracteate, K stands for calyx, C stands for corolla, P is perianth, A is androecium, G for gynoecium, G underlined is superior ovary, and G bar for inferior ovary. This is for female. This is for female. This symbol for bisexual plants and plus in the circle for anti-actinomorphic and percentage is for zygomorphic. Nature of flower fusion is indicated by enclosing the figure within a bracket and addition of by a line drawn above the symbols of the floral parts. The floral diagram provides information about the number of parts of a flower, their arrangement, and the relation they have with the one another, which is located in figure 5.16. The position of the mother axis with respect to the flower is represented by a dot on the top of floral diagram. Calyx, corolla, androecium, and gynoecium are drawn in successive wall. Calyx being the outermost and the gynoecium being the center. Floral formula also shows cohesion and adhesion within the parts of walls and between walls. The floral diagram and floral formula in figure 5.16 represents the master plant family Brassicaceae solanacea. It is a large family commonly called as the potato family. It is widely distributed tropics, subtropics, and even temperate zones, which is located in figure 5.17. Vegetative characters. The plants mostly shrub and rarely small trees. Stem herbaceous, rarely woody, aerial, erect, cylindrical, branched, solid or hollow, hairy and glabrous. Underground stem in potato solanum tuberosum. Leaves alternate, simple, layer, rarely pinnately compound, stipulate, exstipulate, venation reticulate. Solanum nigrum plant, flowering twig, flower, layers of flower, stamens, carpel, floral diagram. Floral characters, inflorescence, solitary, axillary, or cymose, as in solanum. Flower bisexual, actinomorphic, calyx, sepals, 5 united sep persistent, volvate aestivation, androecium, stamens, 5 epipetals, gynoecium, bicarpellary, obligate, placed, syncarpus, ovary, placed, ovary superior, bilocular placenta, swollen with many ovules, axile, fruits, berry or capsule, seeds, many endosperms, floral formula plus in circle, bisexual, ca- calyx, corolla, androecium, gynoecium. Economic importance. Many plants belonging to this family are a source of food: tomato, brinjal, potato, spice, chili, medicine, belladonna, ashwagandha, fumigatory, tobacco, ornamentals, petunia. Summary: flowering plants exhibit enormous variation in shape, size, and structure, mode of nutrition, lifespan, habit, and habitat. They have well developed root and shoot systems. Root system is either taproot or fibrous. Generally, dicotyledonous plants have taproots, while monocotyledonous plants have fibrous roots. The root in some plants get modified for storage of food. In stem, leaves, flowers, and fruits, the morphological features of stems, like the presence of nodes and internodes, multicellular hair, and positively phototrophic nature, helps to differentiate the stem from roots. The leaf is green in color to perform the function of photosynthesis. Leaves exhibit marked variation in their shape, size, margin, apex, and extent of incision of leaf blade lamina. The flower is a modified shoot meant for sexual reproduction. The flowers are arranged in different types of inflorescences. They exhibit enormous variation in structure, symmetry, position of ovary in relation to other parts, arrangement of petals, sepals, ovules, etc. After fertilization, the ovary is modified into fruits and ovules into seeds. Seeds either may be monocotyledonous or dicotyledonous. They vary in shape, size, and period of variability. The floral characteristics form the basis of classification. and identification of flowering plants. This can be illustrated through semi-technical description of families. Hence, the flowering plant is described in a definite sequence. By using scientific terms, the floral features are represented in a summarized form as floral diagrams and floral formula. यहाँ पर हमारा फ्लावरिंग प्लांट्स ख़त्म होता है जिसमें हमने स्ट्रक्चरल ऑर्गेनाइजेशन देखा। सेकंड यूनिट है हमारा सेकंड यूनिट स्ट्रक्चरल ऑर्गेनाइजेशन, जिसमें हमने मॉर्फोलॉजी ऑफ फ्लावरिंग प्लांट्स देखा। मॉर्फोलॉजी ऑफ फ्लावरिंग प्लांट्स में फाइब्रस रूट्स, टैप रूट्स, फाइब्रस रूट्स, एडवेंटिशियस रूट्स देखा। उसके बाद रीजन ऑफ रूट्स देखा, रीजन ऑफ मैचुरेशन, रूट हेयर्स, रीजन ऑफ इलोंगेशन, रीजन ऑफ मेरिस्टेमेटिक एक्टिविटी, स्टेम देखा। उसके बाद लीफ वेनेशन, अरेंजमेंट ऑफ वेन्स एंड वेनलेट्स इन लैमिना, types of leaves, phyllotaxy, inflorescence, flower, parts of flower, calyx, corolla, fruits, gynoecium, seed, structure of dicotyledonous, monocotyledonous and typical formula of Solanaceae family, and summary. तो यहाँ पर हमारा मॉर्फोलॉजी ऑफ फ्लोरिंग प्लांट्स ख़त्म होता है। थैंक यू सो मच फॉर वॉचिंग।